No.
That's the point. I think
Pigeon fancies have changed. That's what's changed.
I think the competition has changed too, because I know that back in the 60s I used to read more about it then and actually participate. But I do know that we had some very good fanciers then, you know, national names in the area. But you saw most of the long races won on the second day. Now today, if you don't get your pigeon on the first day, you've had it, boy. We've got fanciers in our club now that, well, there's one partnership there.
Hello, I'm David Parsons and welcome to my DVD on microscopes, how to use them and examine for canker, coxy and worms. I decided to try and put this into a DVD purely because there's a lot of people that have wanted to come on these courses, but unfortunately you live up in the north of the country and I live decidedly down south. And consequently I thought, well, if you can't come to me and I rarely get the chance to come to you, then perhaps the DVD is the best way forward. This DVD is based on my practical one-day courses on how to use your microscope. It's really been aimed at those that are interested in buying a microscope and also those that have a microscope but really don't know what they're looking at. The aim therefore is to enable you to perhaps select your microscope that will suit you best, use the microscope and take care of it for ones that you already have, help you separate disease organisms from normal contents of feces, familiarise you with the life cycle of these disease-causing organisms, and lastly, to talk a little bit about treatment. Over the next hour or so we're going to be looking at how to use your microscope and how to take care of it, causes of poor image quality, diseases that you can monitor using your microscope, how these diseases spread, some techniques on how to monitor for the coxy, canker and worms, and lastly how to interpret your findings and what to do about them. The DVD is split into 4 chapters. The first chapter is about the use of your microscope and what the bits and pieces are. The second chapter deals with diseases that you can see using your microscope and a little about how diseases spread. Chapter 3 is concerned with techniques and interpretation and control of the diseases that you're monitoring, and then at the very end I've got an extras where I've just put bits and pieces I thought were interesting but I really didn't know where else to put them. This slide lists the various bits and pieces of your microscope. In essence it's split into a light source, a stage in which you can place your specimen, lenses to magnify the image, and eyepieces for you to look through. Right, before we go any further, let's just remind you have a pause button on the control for your TV, and it might be useful to use this periodically. If you wanted to read more that's on the slide, use the pause button. A microscope is either going to be monocular or binocular. It will have eyepiece lenses at the top of the tube and objective lenses at the bottom. The eyepiece lenses are usually ×8 through to ×10 or even wide field ×15. This particular head of a microscope is a binocular one. It is my old microscope, and the eyepieces you can see actually slide horizontally to accommodate the position of your eyes in your skull. The object lenses are at the bottom of the tube, and they're either singular or they can be mounted on a turret so that you can have 4 or more interchangeable object lenses. The length of the lens is usually a good indicator of the magnification, so the shorter the lens, the lower the magnification. The colours enable you to select the right magnification easier. Magnification of your sample is simply obtained by multiplying the eyepiece magnification by the objective magnification. In this case, ×10 eyepiece ×40 object equals ×400. This picture shows us plant cells. Plant cells are usually very regular, can be seen in rafts like this or singularly. Sometimes they're more polygonal in shape, but generally have a relatively thin wall with foamy looking contents. This picture shows you a roundworm egg and a few coccidioidosis at low power, be about ×100 total. Black air bubble smack in the middle. This one is a picture of the same roundworm egg and coccidia, but it will be nearer times 400 in total magnification. Here we have again a low power picture of a hairworm egg, again probably round about times 100 in total magnification, and here we've come right up to times 400 and we can see the detail within the hairworm egg very, very clearly at this stage. This next short video shows you a roundworm egg and some black air bubbles. We're just focusing in and out slightly here. Now we've gone upper magnification, so that was times 40. We've now gone up to times 100, and the somewhat exaggerated magnification that we're getting here is down to the fact that I'm using a digital camcorder on the microscope, and this was we're setting the object lens to times 40. So in theory, total magnification is times 400. But because I've not altered the light source, there's less light getting through now, so the whole image has gone much, much darker. However, at this point you can see the edge of the, or outer wall of the large roundworm egg. It's very thick, and sometimes it's also sticky, so you get a lot of fecal material attached to the outside wall. Drop down the magnification again now, and go down one further, and yet again you can see here the effects of focusing up and down using either the fine focus control or the coarse focus control. Air bubbles are very, very useful. If you focus on the air bubbles when you're using a saturated salt solution, the eggs and the coccidia will float up to be in the same plane as those air bubbles just underneath your cover slip. And effectively what you're looking for here is shapes. That's the first clue you get. So we've got air bubbles there, and what have we got coming up here? Looks like a worm egg, but isn't. Next we move on to the light source. This can either be a bulb or natural daylight. Underneath the stage there's often a condenser, and this is merely another set of lenses to successfully trap the light and shine it through your slide. And this is a sort of figure to indicate just how that light is being caught underneath the slide and focused up to the eyepiece. So this is a relatively inexpensive microscope and very basic microscope. It's got a single eyepiece, monocular, although it's got a turret at the bottom with 3 different objective lenses on it. You can now see the coarse focus, and the smaller knob below it is the fine focus. The stage is underneath the object lenses, and we've got spring clips on this to hold the slide and stop it from moving about. Underneath this we have a mirror. One side is concave, the other convex, and that is used to shine the light into the specimen. The iris diaphragm is used to alter the amount of light that's shining onto your specimen. In general terms, the smaller the aperture of the diaphragm, the greater the clarity of the sample that you're looking at. However, obviously there becomes a point when it gets so dark you can't see anything. The stage is absolutely vital as a support for the slide and the cover slip, and allows you to move the sample around underneath the object lens so you can have a good scan through. This is an older binocular microscope. You've got a turret underneath with swivel object lenses. You've got a stage, but this has got a mechanical stage on it, which is a lot easier to use than spring clips. You'd be amazed how jerky you are when you move a slide around underneath spring clips. Which then comes down into the base, which contains the light source. It has a rheostat to alter the brightness of the light coming in. Then also we've got a digital camera connected to this, and also a computer. Focusing and finding the sample is usually the most difficult part of the job. Usually I start by looking for the edge of the cover slip. This is usually quite black and obvious. This picture shows a roundworm egg and 4 coccidial oocysts at 2 o'clock, 6 o'clock and about 8 o'clock. This egg is slightly out of focus. Now you see it sharpened up. I was using the fine focus control on the microscope to achieve that, but in the process of doing that, the coccidial oocysts have actually gone out of focus. This is a non-sporulated coccidial oocyst. I describe them as an outer shell, a little bit of clear white and a yolky bit in the middle. As they become infective, they divide into sporocysts, and you can see 2 sporocysts here. But you've got to remember that these coccidial oocysts are 3D, they're a bit like rugby balls. And so as you focus down, you focus on different levels within that oocyst. So we have a slightly different view of 2 of the oocysts now, and then as we move along, we come into the what looks like a single oocyst right at the very bottom of the field of focus in this particular instance. The limb of the microscope actually supports all the bits that you need: the tube, the eyepieces, the stage, condenser, and also supplied housing for the light source. This is a more modern microscope. We've got the light source in the base again. It's a binocular microscope. Condenser you can now see just underneath the stage there. Focus control at the base of the limb on this one. Mechanical stage again. And the eyepieces on this particular binocular microscope actually swing out a little bit, the V fashion, rather like a pair of binoculars. Taking good care of your microscope is vitally important. Salt solution is highly corrosive, and this is definitely a case of do as I say, but not as I do. The next couple of pictures show you the results of misuse of salt solution on the stage of the microscope. You can see it crystallizing there, and then when we have a look at the next picture you can see the corrosion, and this has resulted in damage to the mechanical stage and loss of functionality of the movement of the stage. Perhaps the most important things are: keep your microscope dust free and keep it clean. Do not allow any salt solution to remain spilt onto the stage at any time, and most importantly, do not allow the object lenses to come into direct contact with the samples. This is one of those times when the pause button would be very useful, so you can see what possibilities we've got. This is a roundworm egg. That's how we like to see it. If I increase the light so there's much more coming through, you suddenly get this white out. And also we've changed from this sort of oval view to a square view of what's going on. You can see nothing. You don't know where the samples are. You don't know even if you've got anything there. But as you reduce the amount of light coming through, then we can start to see our roundworm egg again. And the more you reduce the light, you eventually come to a point when you have optimum clarity, so you can see the greatest detail. If you close it down too much, it quite simply will just go black. And here again we just focused up and down a bit. Other problems include centering the light source. If the light source and your condensers, iris diaphragm, objective lenses and eyepiece lenses are out of true, then you won't be able to see anything very clearly. Some microscopes even have filter slides in the tube, and if these get knocked you can suddenly find you've got sort of semi-lunar black shapes across your field of view. And it's just again due to the fact that either the object lenses aren't clicked into place properly, or one of the carrier slides is still part way across. Other times you can find that the focus control knob loses friction, and so as you're looking at the specimen, the sample gradually drifts out of focus as the stage drops. Jagged lines, you'll see these on a reasonably regular basis as you manage to crack cover slips by pressing the object lens onto them too hard. And lastly, you may also sometimes see moving wiggly lines across what looks like the eyepiece, and you think maybe you've got worms, but unfortunately these are artifacts that are occurring in your eye and not on the specimen. In Chapter 2 we're going to look at what diseases can infect your pigeon and which of these diseases we can actually monitor using the microscope. And finally we're going to have a quick look at how diseases spread. Diseases can be split into non-infectious diseases and infectious diseases. Of the non-infectious diseases we can look at things like broken legs, egg impactions, genetic abnormalities, poisons, sometimes moss for instance, or metabolic and nutritional diseases. In the latter case, we could be looking at rickets, which I've seen recently, and may become more of a problem now that we're rearing youngsters on the darkness system. Infectious diseases can be caused by viruses, chlamydia, mycoplasmas, bacteria, protozoa, which includes the coccidia and trichomonas, or canker, worms, mites, lice and fleas. Of these, we can only actually see some of them down the microscope. Viruses, chlamydia and mycoplasmas we can't really separate in the light microscope, but we will be able to see bacteria if we use something like a times 400 to times 1000 total magnification. Fungi and yeasts are easier. Coccidia and worm eggs are very easy, and then if you really feel excited you can use low power to look at mites, lice and fleas. This is a high power picture of a chicken's gut contents, just showing stained with a special stain that separates bacteria either into blue, purple staining bacteria or red ones. E. coli and Salmonella would both stain red, and they are generally rod-shaped. And as you can see from this, there are very many different shapes and sizes and colours of bacteria here, and there's absolutely no way that you'd be able to tell the difference between an E. coli and a Salmonella just by looking at them. So let's have a look at the coccidia life cycle. This is a direct life cycle. The pigeon ingests an infectious coccidial oocyst, which releases the infectious stages, and these migrate into the gut cells that line the length of the intestine. They go through several replication stages, which can take between 5 to 7 days before more oocysts are produced. As a result of this, the oocyst is then shed in the droppings. This picture shows two non-infectious unsporulated oocysts. One of them happens to be rather large and oval, whilst the other is more spherical. There are in fact 3 different species that infect pigeons. So what are the important points to remember about coccidia? Well, firstly, the oocyst that is shed in the feces is not infectious. In other words, a pigeon could eat it and it wouldn't come to any harm. These oocysts are extremely tough. They can persist in the environment for many months, but under ideal conditions they can actually become infectious within 24 hours. When this happens, it requires warmth and moisture, and one oocyst can give rise to somewhere between 100,000 to 1 million progeny. Whilst the build-up of infection can be extremely rapid, you must remember that the anticoccidials that you might give your pigeons will not kill these oocysts. The coccidia in the gut are going to cause damage to the gut cells. This will allow fluid and proteins to leak from the body of the bird out into the lumen of the intestine. It will interfere with the ability of bird to absorb and digest food, and it will alter the normal protective bacterial gut flora. And if the worst comes to the worst, pathogenic bacteria may even get through into the bloodstream. Essentially, there are 2 types of life cycle for worms, direct or indirect. The large roundworm has a direct life cycle. The female lays eggs into the gut lumen. These eggs are passed out in the droppings, and providing the conditions are right, will mature to contain infectious larvae. This infectious larvae within the egg, or sometimes if it is free of the egg, then is consumed by the pigeon, and from which the larva then matures through a couple more juvenile stages before becoming adult worms. These next 2 pictures are high-power photographs of the large roundworm eggs, and at this point they are non-infectious. They do not contain an infectious larva. The indirect life cycle is very similar to that for the direct, the difference being that a second host is required for the completion of the life cycle. This life cycle is most commonly seen in tapeworm infections, but also occurs in some hairworm infections. The intermediate or second host can be a slug, snail, earthworm, beetle or fly. The egg is eaten by this second host. The egg matures to contain an infectious larva, and when the second host is eaten by the pigeon, it then breaks out and infects the gut of the pigeon. This next few pictures shows us the variations you may see in the common hairworm or capillaria eggs in feces samples. So this is a brownish one. Here we have a nice dark air bubble with a white centre and a hairworm egg about 2 o'clock on top of that. And if we move on to this next side, we now see a browner and darker hairworm egg, which has actually started to divide. You can see the individual cells within it. And this next one is a green worm egg, and this, I believe, is just quite simply due to the presence of bile staining. This short video sequence shows what looks like a hairworm egg there at about ×100 magnification. Not in focus very well. We've got plenty of air bubbles here, so we know we're in the right plane. There's another shape that could just be a hairworm egg. At this stage it's still not very clear. A bit more light doesn't seem to help in determining whether or not this is in fact a hairworm egg. So we increase the magnification, so we're going up to ×400. The problem now is we've lost our potential hairworm egg, so you've got to have a bit of a hunt around. One air bubble, not far away. There we go, and there we can see in better detail that yes, this is most definitely a hairworm egg. Lemon shape, 2 bottle stops at each end, and you can start to see some of the structure inside. So now some important points about hairworm and roundworm infections. Firstly, one worm, female worm, can lay many hundreds of eggs, and there's no direct correlation between the number of worm eggs you see in the dropping and the number of worms that will actually be in the gut of the bird. Next, it's important to remember that juvenile stages don't lay eggs, but they will still be there. This is a so-called pre-patent period, that time between when the egg is ingested before we actually see eggs being laid in the droppings, and this can vary. The eggs can persist for many months in the environment and cause trouble later on. They develop into infectious larvae quickly when there's warmth and moisture available. Whilst you might well see the large roundworms shed in their feces, you're less likely to see the hairworms, although you can do this if you happen to put the droppings into a pot, and you may just see the worms swimming to the top. This next short piece of video shows worm larvae in a pigeon dropping. The only time I've actually seen these, and they're quite rare, usually at the Blackpool Pigeon Show. These particular worms are Strongyloides species, and they're unique in as much as you have a free living stage, which is in the droppings, and where you have both the male and the female worms, and also an infectious stage. The infectious stage is caused by the female worm only. This is one of these unique little adaptations that Mother Nature has come up with, but in essence, again, the female worm in the pigeon's gut is going to be causing some quite significant damage, which at the end of the day could well debilitate the bird, result in loss of body weight, certainly affect performance. But fortunately for us, these stages are treatable when they are within the pigeon. However, as you can see, it's really perhaps one of the more fascinating little oddities that you'll come across every now and again, certainly not regularly. We now move on to the life cycle of feather lice. This picture shows both the slender feather lice and the fatter, shorter body lice. The life cycle is completely on the pigeon. The eggs are attached to the feathers at the base generally. From these then mature, and from these hatch nymphs. There are 3 nymphal stages before they become adults, and the whole of the cycle can take around about 21 days. Generally, the feather lice don't cause too much problem because they're only feeding off feather and skin dust and dander, although sometimes the body lice may suck blood. This is a picture of the pigeon lousefly. They're flattened top to bottom, and they can scurry about through the feathers extremely quickly, and if you try and catch them, they sort of fly off rapidly and are very difficult to pin down. They suck blood. They have a very interesting life cycle as well, in that the larva actually develops within the female, and when it's passed by the female, rapidly pupates in crevices before hatching, and this can take between sort of 23 to 31 days. Every now and again, when you're looking at your feces samples, you may come across these mites. The problem we have then is deciding what sort of mites are they. Have they just come from the feed? Have they come from the environment? Dust mites as opposed to anything else, or are they indeed feather mites that have dropped off and then been either just landed in the droppings or been preened off by the pigeon? I must admit these days I tend to work on the basis that they could well be feather mites, and that perhaps the safest course of action is actually to treat for them. Now let's have a quick look at the life cycle in this instance for the red mite, because we do actually know a bit more about this than many other mites. The life cycle can be completed in a shorter period of time as 7 days. The eggs are laid by the females in cracks and crevices, and these then mature to give rise to larvae, which don't feed but molt rapidly to become nymphs. The first and second stage nymphs do require a blood feed before they molt eventually to become adults. The adults basically live in cracks and crevices within your loft and only come out at night usually to take a blood feed from the pigeon. There are very many species of mites that can affect pigeons. Some cause damage, such as the depluming mites and the quill mites, whereas others seem to cause very little damage at all. But the life cycle in general will be similar to that of the red mite, but in this instance the whole of the life cycle tends to take place on the pigeon. Again, canker tends to have a direct life cycle. The organism multiplies in the throat or in the intestine of the bird, so that the major source of infection occurs when the adults feed the youngsters with crop milk, but infection can also occur through the drinking water, where the organism can survive for a limited amount of time. This is a picture of canker as you would see it down the light microscope at times 400 magnification. On the right we have 2 large cheek cells, and these are what I normally use to focus on, and then on the left hand side we have a little bunch of 4 or 5 canker organisms. The giveaway is that they always move. If you make an air dry smear from the throat of a pigeon, you may be able to see something similar to this. This has been stained with Diffquick, and you can see a trichomonad, sort of top left hand corner, sort of pear-shaped organism. You can just make out 2 flagellae, little arm-like things, coming off the top pointed end, and then down below we've got cheek cells which are covered in a mass of darker blue staining rods. So lastly, let's just quickly go through how disease is spread. Well, as far as worms and coccidia are concerned, it's through pecking and eating of droppings. If you're tapeworms and some other roundworms, then eating slugs, snails or earthworms are a good way of picking it up. If it's canker or bacteria or yeast you're worried about, then the drinking water can also be a good source of infection. Poorly healed navels in newly hatched chicks can acquire canker if they happen to be in a dirty nest. Crop milk is obviously also a good source of canker, and then there's a mechanical transfer of disease on hands, dirty overalls, shoes, or quite simply just the introduction of new birds. In Chapter 3 we're going to look at the techniques involved in examining dropping samples and throat swabs. This will include how to prepare your fecal flotation solution, how to interpret your results of your droppings examination, and possible treatments and control. But firstly, a few simple warnings. All your samples need to be put on a glass slide. If you decide to reuse your glass slides and wash them, be very careful to avoid serious cuts. Never wash cover slips, but do dispose of them carefully so that you don't cause potential damage to other people. Never place the object lens in direct contact with the sample. You should always have a cover slip on top of your sample, the only exception being if you're doing oil immersion work. Remember, salt solution is highly corrosive, so keep everything clean. Preparing a salt solution is very simple. You just need an old saucepan, a wooden spoon, and water and salt. Add 1 or 2 litres of water to the pan and bring to the boil. Then keep adding salt until you can dissolve no more, and eventually a white crust will begin to develop around the top of the saucepan. When you reach this stage, turn off the heat and let it cool down. Remember, this water will be boiling at higher than 100 degrees centigrade. It is superheated water and can cause some very, very nasty burns. The water is boiling, and whilst you can see some salt having crystallised around the top, the bulk of the water is still very clear, so you can keep on adding salt at this stage. Eventually, when you add more salt to the water, it does not dissolve. At this point, turn off the heat and let the pan cool down. You will see that you have salt precipitating to the bottom of the pan, and you'll also have salt crusting around the top. But this is our saturated salt solution. Just put it into a suitable plastic container. So let's slowly work our way down the pigeon from the head end. Throat swabs first. Place a couple of drops of warm room temperature water on your glass slide. Dampen a cotton bud and take a sample from the back of the throat, or if you like to go a little bit further down into the crop of the bird. This is easily done just by simply running the swab along the roof of the mouth. Then you have to go down into the throat. Bring the swab out and roll it in the water on the slide. Then place a cover slip on top of this. Do this by simply placing one edge of the cover slip at the edge of the water on the slide to give you about a 45-degree angle, and then just slowly lower, trying to avoid the production of too many air bubbles. Canker is the only large moving organism that you will see in this preparation. So, potential problems with throat swabs. Firstly, cold water may kill or, at the very least, stop canker from moving. If it doesn't move, you don't know it's there. In some areas, there's so much chlorine in the water that the canker may die quite rapidly. I've tried using bottled water in these instances. If you leave a canker for too long in tap water, it's a bit like you sitting in the bath. It just simply swells up, but in their case, eventually dies. And whatever you do, do not use flotation solution to look for canker. When looking for canker, you may well see other things under the microscope. We're using a ×40 objective, so it's quite high powered. You'll see the cheek cells easy enough, and then every now and again you will actually also see some budding yeast cells. These are single cells but highly refractile. Okay, now for looking at our feces flotation test. Firstly, collect about a dozen droppings from a section or the loft. Put them into a plastic bag or a suitable container and give them a good stir, so you mix everything up. Take some of this and place about a quarter to a third of feces into a fecon tube. These are 27mm plastic tubes. Add some saturated salt solution to cover the droppings and give it a gentle stir. We don't want to have too many air bubbles. Having done this, then add more salt solution till you bring it right up to the lip of the container so that it just sits proud. Next, carefully lower a 22 by 22mm square cover slip onto the proud surface of fluid at the top of this tube. Again, try not to cause too many air bubbles. The coccidia and the worm eggs will then float up over time and stick to the underneath of the cover slip. I generally tend to leave samples to float for about 30 minutes. After this time, you then carefully remove the cover slip, taking with it a little bit of water, and place this on a glass slide. Again, put one edge to the slide first, sort of at a 45-degree angle, and carefully lower the remainder of the cover slip onto the slide, again trying to avoid too many air bubbles. Next, place the slide on the microscope stage, and with a 10x objective, total magnification about times 100, look for the edge of the slide, and then I tend to try and aim for one corner. Having found that, I then focus on air bubbles and slowly but surely scan the whole of the cover slip. Scoring your results is relatively easy. Just use the tables here as a rough guide. It's very similar for hairworms and for coccidia, and perhaps the easiest thing to do is just press the pause button on your remote controller so you can take in exactly what I've got written down here. The scores run from 0 to 3. 0 means you don't see anything, and 3 means you see rather too many of everything. The interpretation of the results really depends on what's going on with the birds at the particular time. For instance, I generally like to see a score of 1 for coccidia in birds most of the year, but not when they're racing. In contrast, if I see hairworms at a score of 1, I want to treat immediately. Roundworms I may be a little bit more relaxed about. This slide tells you how I tend to interpret the results that I find. However, it is vitally important to know what is going on. Are they young birds? Are they stock birds? Are they old birds? Are they racing birds? All of these things make a difference to the interpretation and consequent treatment that you may or may not wish to undertake. Every now and again I'm sure you'll send samples off to a lab and you'll come back with numbers rather than scores, and I put this table in purely to give you, again, a bit more of a clue as to how you might possibly wish to interpret those results. Just press the pause button so you can look at it a bit longer. There is absolutely no reason why you shouldn't look at a feces sample directly under the microscope. The important things to remember here is, are you going to use water or are you going to use salt solution? If you use salt solution, then certainly you will kill off any hexameter and trichomonas. If you use ordinary plain water, then you may well see hexameter and trichomonads if they happen to be there. But another most important thing is, do not take too much sample. If you make it too thick, then the simple thing that will happen is you'll see nothing at all. You can also take smears from the cleft in the roof of the mouth of the pigeon and also from the vent. To do this, just moisten a swab, either in normal saline or tap water, and place a drop of water or normal saline on the glass slide. Having taken your swab from the vent or the cleft, then carefully roll the swab in the water on the glass slide. Spread this around to give you about 2 to 3 centimetres square at one end of your slide, and let it there dry. Then I simply stain with either Diff Quick or Rapidiff stains. Again, allow them to dry and examine by oil immersion, in other words, times 1,000. These next few pictures may give you an indication of what you might see. This is an impression smear from a duck, and what we've got here are basically some large purplish looking cells and some bluey rod type cells. The important things that we're really looking for here is, have we got any inflammatory cells? And in this picture you can see 3 large circular cells which are inflammatory cells with the red granules. The remaining cells, oval shaped with the blue centres, are in fact pigeon red blood cells. A similar one here, but in this case instead of red dots it looks more as though we've got red granules. Perhaps I should have talked about collecting samples before we started, but at the end of the day you've got to make a very simple decision. Is this routine or is it a disease investigation? If it's a disease investigation, then we may be just looking at 1 or 2 birds, in which case we want to collect samples from them. If it's a routine test, then are we going to check the whole loft, say at the end of the molting season, or are we going to be checking the race teams, or are we going to be checking the young birds? In these next few pictures I'm using a fecon pot to collect sterile samples with the enclosed spoon, and I'm just picking the odd dropping up from different box perches off the floor, making sure that I only hold the top, don't put my fingers inside. I'm not interested in sampling myself for bacteria, just what's in your pigeons. And you only need about half a pot full, so that's going to boil down to about 6 to 8 droppings from any one section. this is quite simply just a close-up to show you how much we actually need from any one group of birds. These next few pictures are of different types of droppings that you can see, but just by looking at them you really don't know whether it's young bird sickness you've got, circovirus, adenoviruses, paratyphoid. This one happens to be paratyphoid because I can remember taking the samples and getting the positive result, but just by looking at it you wouldn't even know whether it was paratyphoid or paramyxovirus. This one, I think if my memory serves me right, was possibly associated with hexameter. And when you come on to look at this particular bird and think, well, okay, it doesn't actually look too bad like that, but it was one with young bird sickness, and it happened to be having some of the most awful diarrhea that you could ever wish to see. Extremely sloppy, but these are the ones you could take just a direct smear from. So how do we control our intestinal parasites? Well, environment and management are vitally important. Try and keep the birds separate from their faeces, using grids or regular removal of the droppings. Don't feed on the floor. Keep the loft dry. Use loft powders, Lysan, Stalosan F, and perhaps every now and again disinfect with oocide or kill cocks. Regular monitoring of your birds is essential if you're to try and build up a picture of what is actually going on, and then based on these results you can institute a specific program to achieve the best results. And the very simplest it is, has the treatment actually worked at all? You are actually spoilt for choice with treatments these days, and whilst there may be certain fads in certain areas, there is a readily available supply now of coccidiosis treatments, worm treatments, and feather lice and mite treatments. It's not perhaps so much a case of do the treatments work, it's more a case of are you actually giving them at the right time, and the assumption that you only need to do it once a year is totally fallacious. A few years ago I had the opportunity of comparing an ivermectin spot-on with Avicas to control worms. So we had 4 sections. We divided 2 sections onto the ivermectin and 2 sections onto the Avicas capsules. We took samples of droppings before we treated, and then we gave the appropriate treatments. Avicas given on one day and the ivermectin drops given once a week over 3 consecutive weeks. Then 7 days after the last treatment we had another look at the droppings. So what were the results? Well, didn't have any worm eggs in the Avicas treated birds before treatment, but we did find hairworm and roundworm eggs after treatment. With the ivermectin sections we found both hairworms and roundworms before and after treatment. So what is this suggesting to us? Well, firstly, the failure to find worm eggs doesn't mean there's no worms, it just means that there are juvenile stages there which perhaps your wormer will not be able to treat. Ivermectin spot-ons, at least at the levels that I was using, don't seem to act as wormers, although they do act very well as mite and lice control agents. As a result, I would suspect that the Avicas treated birds did in fact have juvenile stages in them, and this is a very good reason for why monitoring is so essential. So what did we do? Well, we treated for worms again, then we checked the droppings again 1 week after the treatment had finished, and that worked fine. However, in this instance we weren't using the ivermectin, but this does raise the question whether there was sufficient ivermectin in concentration in the drops that we were using to both act as a lice control product and also as a worm control product. Well, here we are at the end of this DVD. These are my contact details should you wish to get in touch with me at a later stage, and may I at this time thank you very much for taking the trouble to A, buy the DVD, and B, I hope that getting to this stage also means you've enjoyed what I've had to say or show you. Lastly, may I thank all the fanciers that have provided me with continuing stimulus to learn more about pigeon pathology and parasitology in particular, to Andrew Parsons for doing the graphics and production, to the voices of Jason Edwards, Owen Elliott and Frank Quinn, and to Brunel Microscopes for supplying some excellent equipment and advice when needed. So here we go, the extra pictures. This first one is going to be of a roundworm egg. rather thick smear, so it makes the whole thing look rather unclear, but you can still see the very thick wall. The next picture shows this much clearer, very clear wall, but these at this stage are non-infectious. The larvae has not developed. For those of you that look at chicken droppings, this is a picture of a heterachus egg. It's very similar to a roundworm egg, except for it's not quite so square or brick-shaped. It has thinner walls, although it's probably 3/4 to 7/8 the size of the roundworm egg. These are particularly important in chickens because they live in the blind gut or the ceci of the chicken and can be involved in conditions such as blackhead. Other infections we sometimes see in pigeons are supposedly gateworms. I've not seen it in the UK, but certainly it's written about in the books. This is an out of focus picture of a gateworm egg, and here you see the same thing but in focus. And already this egg, the contents have started to divide to give us several cells rather than one. This is a reminder of an air bubble and a hairworm egg. In this case it's brown, but in other cases it can be green. And alongside this one we've got a coccidiolysis at about 3 o'clock. And from this you can see that coccidia are approximately a third the size of a hairworm egg. The reason for the green is, I believe, bile staining. These are tapeworms. They're taken from a chicken, but I just put them in to show you the sort of segments that you can see, and I know this was a still photograph, but at the time the segments were still moving. These can be particularly vicious and nasty infections. This is just to show you a whole selection of different air bubbles, and they can be all shaped, well, generally round, sometimes all different shapes. They always have a clear middle, and the one at the top right hand corner almost looks as though it has an internal content, which can cause some confusion occasionally. These following slides are actually of ground pigeon corn. This is barley, and I did it so that I could attempt to recognise bits and pieces that you might well see when you're looking at a dropping sample, to try and ensure that I didn't mistake normal for abnormal. This is paddy rice ground up, and it's darker and more homogeneous. This is maize, and you can almost see the granules here. And this is pelleted feed. It's actually very difficult to make any sense of what you're looking at all. But it's certainly been ground up nicely. This particular picture shows you a raft of plant cells, and again to remind you, they tend to be more regular, very often square, but otherwise polygonal, and very distinct edges. This is a picture of a maize cell with starch granules. If you used iodine, even Lugol's iodine, you would then see that these granules start staining blue. They have been mistaken for worm eggs. This video shows us, again, some starch granules, and here we're hunting around, focusing primarily on the air bubbles to see if we can see anything else, and it's the shapes. Always you're looking for shapes, and here we've got something that looks like a hairworm, but really you can't quite make it out at this stage. Go to a higher power, and then if you're lucky, your microscope will be parfocal, which basically means that when you change to a higher power you should be over the object in question, and now you can begin to see the distinct outline of the hairworm. If we go higher power still, and using the digital camera on this microscope means we do go very, very high. It also has gone dark, and that's because I haven't got enough light coming through the condenser. But now again you can at least see the structure of this hairworm quite clearly. The focusing up and down just serves to remind us that these are 3D structures, not just 2D. This is another short video, again just showing us maize cells with starch granules in them, and you can just begin to see now how easy it is to be confused that they might just be something nasty like worm eggs. They're not. This is all on quite low power, times 40 total magnification. This long thing looks like a worm but isn't. There's some spiky bits there that look as though they could have been the ends of mites. They're not, they're plants. This next slide shows you a feather in the dropping sample, and the other dark rings again are air bubbles. But it's important to be able to spot these things and know what you're looking at. This year I actually saw some pollen grains in some samples. Initially I didn't know what they were, so I drew them, and I would suggest that if you see anything you don't know, if you can't photograph it, draw it. At least then you can take it along to people and say, well, what do you reckon this is, and take it from there. Your drawing may not be particularly brilliant, but at least at the end of the day it will give you a good way of describing what you were looking at. These are salt crystals. I really don't like shaking up my salt solution before I use it. I just use the clear fluid off the top. That will work perfectly well. These next two pictures simply show cover slips on top of samples that are being prepared. I found that the 22mm square cover slips sit quite nicely on top of a universal or fecon pot. You just got to sit and wait for half an hour while the coccidia and the worm eggs float up to the top. Unfortunately, it is not possible to tell the difference between E. coli and Salmonella just simply by looking down the microscope. Even in the lab it can be difficult. This is E. coli on a blood agar plate. This is Salmonella on a blood agar plate, or paratyphoid. I defy you to tell the difference between the two. Consequently, we use what is known as selective media. This is a brilliant green plate, and on this Salmonella grow pink and E. coli's grow a yellowy colour. This is E. coli being grown on a McConkey plate. In this instance, the E. coli is red and Salmonella would show up as a pale beige, white, grey colour. This next picture is yeast, been stained with what we call a gram stain, and has come out as looking like a solid series of blue dots. This picture is of an inflammatory cell taken from a blood smear, and that's the one with the red dots at around about 9 o'clock in the picture. There are also 3 blue staining lymphocytes, which are also immune cells, and a series of oval red cells with blue centres, which are red blood cells. In this one we have red blood cells, 2 blue lymphocytes, and 2 inflammatory cells with red granules in them. If you see these inflammatory cells in swabs from the throat or from the vent, then at least you know you've got an infection. Similar picture again, in this case about 2 o'clock top right is an inflammatory cell, and the rest are all red blood cells. For information, the red blood cells of birds have a nuclei, ours don't. And this is an impression smear taken from an infected hock joint, and you can see a red blood cell in the middle and 3 inflammatory cells on the outside. This indicates that we definitely have an infection. And lastly, a little quiz. What can you see in this picture? At least they're all numbered, and now's the time to hit the pause button. Okay, so you didn't keep the pause on for very long. The top left, figure 1, is a hairworm egg, an air bubble. Number 2 is a green hairworm egg with the coccidial oocyst. Number 3 is a picture of cheek cells on the right, and canker cells or canker organisms about 9 o'clock on the left. To the left we then have a large brown hairworm egg. Bottom right we have 2 coccidiocysts. Middle bottom centre, number 6, we have a roundworm egg. To the left of that we have another coccidiocyst, and in the bottom left hand corner we have yet another hairworm egg.